And with that, it is my privilege to introduce our speaker today, Dr. Lorenzo Cingolani. He earned his undergraduate degree in molecular biology from the University of Pisa in Italy. He then pursued a Ph.D. in neurobiology at the Humboldt University in Berlin, specializing in electrophysiology at the Max-Planck Institute for Experimental Medicine. His postdoctoral fellowship at the MRC Laboratory for Molecular and Cell Biology at the University College London led to groundbreaking discoveries in synaptic physiology, emphasizing the biomechanical properties of the brain in health and in disease. In 2012, he established a laboratory at the Italian Institute of Technology in Genoa, focusing on the molecular basis of functioning brain connectivity, which has implications for treating autism, epilepsy, and ataxia. Dr. Cingolani's work has involved advanced techniques like optogenetics and genome editing. Since 2019, he has been an associate professor in physiology at the University of Trieste, directing the Laboratory of Synaptic Neurophysiology. So thank you so much for joining us today, Dr. Cingolani. The floor is yours.
Thank you so much for your warm introduction. I'm very excited to have this opportunity to discuss with you the research conducted in my lab, but also in many other laboratories. And I will focus on preclinical studies, disease mechanism, and research treatment for episodic ataxia. Now, clearly, in episodic ataxia, we have spells or episodes of poor coordination and imbalance, and this really can last from several seconds to several hours. And many of these forms of episodic ataxia can be progressive, so they get worse with age. This is a rare form of ataxia. The incidence is just 1 person per 100,000 people. And these are genetic forms of ataxia. At least I will talk only about genetic forms. And they've been described at least 8-10 different types. And as you can see, this is a table from a recent, this is a table from a recent review where you can see these 8 or 10 different types of episodic ataxia. But some of them have been described only in very few patients, and the two most common forms of episodic ataxia are the type 1 and type 2, with the type 2 being slightly more common and I would argue also more severe. Now, both types of episodic ataxia are classical channelopathies. So they are caused by mutations in ion channels, specifically KCN1 for the type 1, and CACNA1A for type 2. And as we will see, ion channels are proteins which are essential for the excitability of the brain. And the reasons why mutations in this channel induce episodic ataxia is because both of them are highly expressed in the cerebellum. And the cerebellum is this brain region that you see here, which we have here in the back of our brain. And well, this is basic physiology, but perhaps it's important to remember that the action starts in the motor cortex. The information travels down through the spinal cord, and feedback information can travel up to the cortex. And the cerebellum essentially has an optimal position to monitor both this downgoing and upgoing information and to detect whether there are mismatches and compensate for these mismatches. So that if there are mutations in these two channels, the cerebellum doesn't work properly, and this activity of correcting for mistaking is not present anymore, and forms of episodic ataxia may arise. So I will talk first briefly on episodic ataxia type 1, and then I will move to episodic ataxia type 2, where we also performed some basic research. Both types of episodic ataxia normally arise in early childhood, and the type 1 is characterized by brief episodes, so they last typically seconds to minutes, and often they are associated with slurred speech and also with tremor. And well, the frequency really can change considerably from patient to patient. And both type 1 and type 2, the episodes can be triggered by stress. Now, one characteristic of type 1 is that very often it is associated with myochemia, that is with twitching of the muscles, which can be detected, clinically detected, or you need also eventually electromyography to observe. Now, for both forms of episodic ataxia, there are medications that can be used to manage these forms of episodic ataxia. For example, for type 1, the most commonly used is carbazepine, which you can see depicted here. There are also other drugs. Now, the problem is that the response with patients can be highly variable. Resistance to drug can develop, and also side effect can develop. But really, I would argue that the major problems with these drugs is that they've been identified through a process of trial and error in patients. And they've been developed to treat mainly the symptoms rather than the genetic cause. So, and the major aim today for me would be to convince you that preclinical studies, as we will see across different levels, they can lead to the development of targeted drugs or eventually genetic therapies. They can really address the genetic cause of the disease rather than the symptoms. And therefore, they can have a greater potential to be more effective and also with reduced side effects. Now, this brings us to the pathophysiology of episodic type 1. This is an autosomal dominant disorder, dominant meaning that you need the mutation in just one of the two copies of the gene to eventually develop episodic ataxia. It is autosomal, so not strictly linked to males or females. It is, as we said before, monogenic, so mutations in one single gene, KCNA1, can cause this particular type of episodic ataxia. And this gene encodes for a voltage-gated potassium channels, Kv1.1, that you see depicted here. And really, all potassium channels generally, and Kv1.1 in particular, their function is to reduce the excitability of the brain. So that if there are loss of function mutations in this channel, the effect is increased in brain excitability. Now, an important point to consider is that although the mutation, the disease is monogenic, there is not a single mutation, but there have been identified at least 63 different mutations, which are spread out along the channel. All of them are loss of function, and all of them can cause episodic ataxia type 1. Clearly, not all mutations will affect the channel to the same degree, and this can explain, at least in part, the variability that is observed between different patients. Now, once these mutations have been identified, the first thing that normally is done is to investigate whether they indeed are pathogenic, so whether they change the activity of the channel. And this can be done quite easily, for example, by taking the mutated channel and to express it in a cell that normally does not express this channel. For example, an oocyte from this frog. And with standard electrophysiological techniques, it is possible to record, monitor the activity of these channels. And I hope you can appreciate that if you express a wild-type channel, that is a channel naive without mutations, the currents that we can record is quite large, whereas if we express a channel that carries a single mutations, of the many that can be present, the currents that we record is quite reduced. Now, with this very simple approach, we can not only investigate the function of the channels, but we can also investigate whether drugs, which are specifically designed to target these channels, can rescue the effects of these mutations. Now, here, unfortunately, it's not as clear as in the previous slide, but on the left, you have currents produced by a channel which carry one mutation. On the right, you have currents produced by the same channels, but upon applications of this niflumic acid. Now, if you stimulate the channel with 40 millivolt, in this case, we have no current whatsoever, the same stimulus, minus 40 millivolt, will produce a tiny current in the presence of niflumic acid. If we increase the stimulus, minus 20 millivolt, the current increases, but with niflumic acid, it becomes even bigger. So essentially, we can, in this way, we can screen the efficacy of drugs, whether they can rescue and compensate for specific mutations in this gene. Now, unfortunately, this approach is absolutely not enough for brain disorders, and it is necessary to investigate the effect of the mutated channels in the context of the brain region that is involved in the disease. And in this case, as we said, the cerebellum. So it's very important to investigate how the mutated channel can affect both the structural and functional connectivity and the communication between neurons. Here you see a section of the cerebellum. The major output of the cerebellum are the Purkinje cells, which are these beautiful cells with this very large dendritic tree that you see in gray, and they send an axon outside of the cerebellum, so that all the computation that this cerebellum performs is funneled inside these Purkinje cells, and then they will send information outside to other brain regions. Now, the Kv1.1 channel is not expressed in Purkinje cells, but is expressed in these small cells, basket cells, and these are inhibitory neurons. That is, these are neurons that release an inhibitory neurotransmitter, GABA, which inhibits, keeps at bay the activity of Purkinje cells. Now, if we have mutation in Kv1.1, what happens is that this inhibitory effect will be increased, so that we are going to reduce the activity of Purkinje cells. And as a consequence, we will have defects in motor coordination. And now what they did here, just to give an example, the researcher investigated exactly the connectivity between these two neuron types, the basket cells and the Purkinje cells. So they could use an electrode to stimulate the basket cell and a second electrode to record the inhibitory synaptic currents in Purkinje cells, which are displayed here. And here really you need to believe me, these are inhibitory synaptic current, but hopefully you can appreciate that if you apply this drug, niflomic acid, you can considerably reduce this inhibitory current on Purkinje cells. Now, is this relevant for the activity of the Purkinje cells? Yes, it is. And to study this, we need to use mouse models, essentially mice that express exactly the same mutations that have been identified in patients. For example, in this case, we have a mouse which expresses this V408A mutation. And again, if we record with an electrode the activity of Purkinje cells in wild type, so in naive mice which have no mutations, we notice that the activity in Purkinje cells is very regular. So each one of these vertical lines, this is really an electrical impulse. And as you can see, they come at a very regular pace. If you do exactly the same recording in the mouse that carries the mutation found in patients, the overall activity does not change, but this time the activity is much more irregular. Sometimes the electric activity is low, sometimes it speeds up, then it becomes slow again. And again, if you apply this drug, niflomic acid, on the mutant, we can essentially rescue to a big degree this regularity. So hopefully you can appreciate that these Purkinje cells now fire as regularly or nearly as regularly as the wild type Purkinje cells. So what these experiments tells us that we can use a drug which is specifically designed to rescue the mutations in the K1.1 channels. We can decrease the inhibitory input on Purkinje cell, and as a consequence, we can normalize the electrical activity of Purkinje cells and presumably also the motor coordination of these mice. And to see whether we can indeed do this, we clearly need to perform behavioral experiments in mice. Now, here you see a wild-type naive mouse that does what it likes most to do, to run on a beam. And you can see that it does it very skillfully. A knock-in mouse with this mutation would like to run on a beam, but essentially you see that has severe problems in performing this apparently simple task. And I do not have a video to show you the rescue experiments, but from the quantification, you can see that in the presence of the mutations, many more errors are made, and niflomic acid can also at least partially rescue this behavioral performance. Now, I went quite fast, but I wanted to show essentially how research can be conducted on episodic preclinical research can be conducted for episodic attacks. Now, if we put all of these together, what we have seen is that we start from the mutation in patients, a potential pathogenic mutations, to decide whether it is indeed pathogenic. Essentially, we need to investigate their effect on the function of the channel. Does it increase the activity of the channel? And in this case would be a gain of function mutation, or does it decrease it? If there are some effects, then it is very useful to develop mouse models, essentially mice that carry the same mutation that have been identified in patients. And I stress this point again, for brain disorder, it's absolutely essential to investigate the impact of the mutation at the neuronal network level. So how the mutation change the structural and functional connectivity between neurons and between brain regions. And the ultimate test, of course, is to see whether the mutation reproduces the same or similar ataxic behaviors as observed in humans. Now, at each one of these steps, rational drugs, so drugs that have been developed not because they cure the symptoms, but because they can compensate for the causative mutation, these drugs can be tested and screened at each one of this level of investigation. I hope this was clear, and I would like now to move to episodic ataxia type 2, where we also did some research. Now, in the case of episodic ataxia type 2, the episodes or spells of ataxia typically last much longer. They can last hours or in some cases even days. And they're often associated also with vertigo, nausea, vomiting, and in 50% of the cases with migraine. Also in this case, the frequency is quite variable. Stress can trigger these events. And in the case of type 2, very often there is nystagmus, and this can be quite indicative that we are facing episodic ataxia type 2. And as type 1 can be progressive, and in this case, very often there is a degeneration of regions of the cerebellum. Now, as we've seen for type 1, there are drugs, medication that can be used to manage the disorder. And in this case, the most commonly used is acetosalamide. And you can read what is written here, but essentially we do not understand why it works and how it works. And the second medication, which is commonly used, is foraminopyridine, and this is a classical blocker for not the causative mutated gene, but for potassium channels. So also in this case, the medications are clearly drugs that target the symptoms and have been identified through trial and errors on patients. Now, the gene causative for episodic ataxia type 2 is CACNA1A. This is also a voltage-gated channel, but in this case, is permeable not to potassium, but to calcium. The name is Kv2.1, or you will also find picotype channels. And as we will see, this channel is absolutely essential for synaptic communication between neurons. And also in this case, we don't have a single mutation, but more than 100 different loss-of-function mutations have been identified, and some of them are shown in this configuration. And most of them are loss of function, so they impair the function of the channels, and therefore they impair synaptic communication between neurons. Now, for CACNA1A, I think it is quite important to consider this is such an important channel that whenever we touch it, we change it even slightly, many disorders we can trigger different types of disorders. So it is the causative gene not only of episodic ataxia type 2, but there is a second important form of ataxia, spinocerebellar ataxia type 6, which is due to mutations in CACNA1A, different types of mutations. Very often, these forms of ataxia are, you also find cerebellar atrophy, or you find eye disorders. And there are also other disorders which are not necessarily comorbid with ataxia, which very often arise when CACNA1A is mutated. For example, epilepsy, specifically absence epilepsy, migraine, specifically familial hemiplegic migraine type 1, and more recently, cases of forms of neurodevelopmental disorders have been identified. So that this is a quite complex situation where we can have a single mutations that can cause multiple disorders, or different mutations on the same genes that can cause related or unrelated disorders. But very often we find comorbidity between ataxia type 2, epilepsy, and migraine, and sometimes with neurodevelopmental disorders. Now, why this is the case? Why it is such an important channel? Well, there are several reasons. First of all, it is a channel which is expressed everywhere in the brain. Here you see a sagittal section of the mouse brain, so in this direction. And the more red you see, the higher the expression of the channel. And as you can see, it's present everywhere in the brain. And hopefully you can recognize the structure. This is the cerebellum, which is magnified here. And each one of these pairs, each one is a Purkinje cell. So as you can see, Purkinje cells are packed with CACNA1A, so that this can partially explain why, if we have mutations in CACNA1A, ataxic phenotypes can arise. The second reason why it is so important is because it is explained in a very simple way. It is present in nerve terminals. It allows calcium to enter the nerve terminals, and this calcium is necessary to fuse this vesicle, which contain neurotransmitter. Neurotransmitter is then released and can communicate with the postsynaptic neuron. So that if the channel does not work and calcium cannot enter, or not enough calcium can enter, essentially this synapse becomes mute and cannot communicate with the postsynaptic, with the downstream neurons, and the information is, we don't have flow of information anymore. And unfortunately, there are no other channels which can replace CACNA1A if this channel is mutated. Now, one aspect which we investigated extensively and needs to consider when studying mutations in CACNA1A is the process of alternative splicing, which allows a single gene, in this case CACNA1A, to generate not one single channel, but up to thousands of different calcium channels, which we call isoforms. And the way it works is shown here briefly. So you have one gene on the DNA, which produces one RNA, and we can identify on the RNA different regions, which are called exons. And these are essentially spliced together to produce the mature mRNA and the final protein. And for some genes, this splicing occurs always in the same way. But for other genes, like for CACNA1A, it can happen in different ways. So sometimes we keep a specific exon, sometimes this one, sometimes we skip it, or the splicing can occur slightly differently, so that if you consider all the possible combinations, from one single gene, we don't have one, we don't have two, we don't have three, but we can have potentially thousands of different proteins. And they are all similar, but they are different in specific domains, so they perform different functions. So when we consider then the mutations that target CACNA1A, we also need to consider which splice variants are affected, and also if we try to devise genetic therapies, we need to consider which splice variants we need to repair. Now, to convince you that this is really important, I show you a simple experiment which has been performed. So in this case, we use super resolution microscopy where we can see in high resolution the localization of this channel. And we can express one isoform of the channel, which we call A, or a second isoform of the channel, which we call B. And we can see this using fluorescent dyes in green, which is shown here. And we can also stain for MANC13 that you see in this pinky violet color. And this is a protein that is present exactly at the center of the synapse. So if we zoom now in on a single synapse, if we see white, that means the two proteins co-localize perfectly and the channel is exactly where it's supposed to be in order to make the synapse function. If we consider the second isoform, we see that they are not perfectly aligned. Now, these differences are not huge. These are differences in the orders of tens of nanometers. And these are the same distances that we consider if we discuss about whether two proteins or two channels are in direct contact between themselves or they are far apart. And although here we investigate the localization of different isoforms of these channels, clearly mutations in these channels could also affect the localization of these channels and have consequences on the activity of the brain regions involved. Now, how can we study this, whether there are functional consequences? Here you see a scheme which summarizes the working model. Essentially, if we have the A isoform, which is tightly localized with the synaptic vesicles, this calcium let calcium enter, and the calcium can induce the release of the synaptic vesicles because it's close by. And if the channels, as you can see here, there is a certain distance, which doesn't need to be huge. However, it is enough for the calcium not to be able to reach anymore the synaptic vesicles, so this synapse will become silent. This is the theory, so how can we see this? Well, we can use an electrode to stimulate one neuron, and this neuron is in green because we express one isoform or the other isoform of the calcium channel. And we can use a second electrode to record the response in a second neuron. Now, this arrowhead represents the point where we stimulate the system with this electrode. And if we do this, we could have no response, so a flat line, because the synapse does not work, or we could have a response that you can see here. When we express, and this we can, when we express the A isoforms, each time we stimulate, we see a response in the postsynaptic neurons. And the way we can express this, we go and count the failure rate. So how many times the synapse fails to work. And as you can see, with one isoforms, the A, there is zero failures, meaning that it works each time. When we express the second isoforms, the B, you can see that many times, sometimes we have a synaptic response, but many times we have a failure. So we stimulate the synapse, but we have no response whatsoever from this side. And this can be explained with not with a different activity of the two channels, but with a different nano-organization of the channels at the level of individual synapses. And again, in this case, we look at isoforms, but similar mechanisms can be explained, can be in place to explain the differences or the phenotypes induced by some mutations. Now, if we want to study more directly episodic ataxia type 2, we need mouse models. And interestingly enough, for CACNA1A, we have at least 5 different spontaneous mutant mice. So these have not been created, they've been just identified in mice. And all of them exhibit ataxia, and many of them also have absence epilepsy. And of course, we can also use constitutive and conditional knockout mice. Now, we wanted to use a more flexible, a new system to develop the mouse models to target specific brain region. So instead of using these classic approaches, we used the CRISPR-Cas9 genome editing technology, which is based on an enzyme, the Cas9, that you can see here. It can be targeted to a specific region of the genome thanks to a guide RNA that you see here in green. And when the Cas9 reaches this specific region of the genome, it simply can cut the DNA. And normally when the DNA is cut, there are endogenous mechanisms that they repair this cut. Now, if there is no extra DNA, this repair occurs in a random way, so a mutation is introduced. And essentially, this is a very effective way to knock down, to essentially mutate a specific gene. Again, if we want to summarize what I presented, it is quite important, not only as we have seen with episodic ataxia type 1, to monitor the activity of the channel, but even apparently minor changes in the localization of the channel can affect the function of the nervous system. And this can be studied with super resolution microscopy. Also in this case, it's important to develop mouse models. And nowadays we have new tools like CRISPR-Cas9 to develop more flexible and less expensive mouse models. And again, I stress the importance to study the connectivity of the brain. In this particular example, we have seen the connectivity between the cortex and the thalamus. And the ultimate test is also in this case the behavioral readout. And finally, a complementary approach, which is quite common nowadays, is to investigate the behavior and the morphology of iPS-derived neurons, which can be, the iPS can be taken from the patients. Now, I haven't touched directly these aspects, but clearly the same CRISPR-Cas9 technology that can be used to develop mouse models can be used to develop gene therapies that can be tested at each one of these levels. And just to summarize the two parts of the talks, episodic ataxia type 1 and 2, preclinical studies, so we always start from mutations identified in patient, and the first step really it is to investigate whether the mutation is really pathogenic. So if it does something to the either localization or function of the channel. For brain disorders, we need absolutely to investigate the impact at the level of connectivity and, of course, at the level of behavior. And it is equally important to test, especially if we look at targeted drugs or gene therapies, to test their efficacy on human iPS-derived neurons, or nowadays also organoids. Let me just very briefly acknowledge and thank, first of all, the Teleton Foundation, which in Italy and in Europe fund research specifically on rare diseases, so make possible this kind of research, and the members of the laboratory, especially Fanny and Riccardo, and some of the experiments have been performed in collaboration with the Gaslini Pediatric Hospital in Genoa. And I thank you for your attention, and I will be happy to take any questions and try to answer them. Thank you.
Thank you for fantastic docs, Dr. Cingolani. As a reminder, we're going to do questions now. You can drop them in the Q&A window at the bottom of your screen. We'll get through as many as we can. The first question is actually for the National Ataxia Foundation, which I'll go through quite quickly. We have an attendee whose daughter has a mutation in CACNA, or my apologies, KCNA2, and is trying to access ataxia care in the United States because their current doctors aren't super familiar with ataxia. We're going to have one of our staff members drop a link in the chat to our Ataxia Centers of Excellence, where you can find doctors who are familiar with ataxia, both in the United States and Canada, as well as elsewhere around the world. So hopefully that will be helpful. All right, so our first question for you, Dr. Cingolani. We have an attendee who's interested in learning more about 4-aminopyridine for episodic ataxia type 2. Could you talk a little bit more about that?
So 4-aminopyridine is a classical blocker of potassium channels. So the idea behind the use of 4-aminopyridine is that if we have a reduction in the activity of these calcium channels, if at the same time we reduce calcium channels excite the neurons, potassium channels reduce the activity of the neurons. Now, if we have a reduction in calcium channels, at the same time we reduce the activity of potassium channels, maybe we can find, again, a balance. But again, of course, it can be quite effective for some patients and very often needs to be titrated on individual patients and also changed then from time to time, updated. But again, it is not be used because it targets the mutated channels. The rationale behind is that we need somehow to re-equilibrate the excitability of the brain.
So what I'm hearing is that there's certain medications that are treating the symptoms and there's ones that are treating the cause, and that we need to invest the time and research into those treatments that are focusing on the cause because they might have better effects long term.
Yeah, this is what I would argue strongly, yes.
And then we have someone who shares that they have EA1, myokemia, and hypomagnesemia, all caused by mutations in KCNA1. In your research or research that you're familiar with, is there anything known about magnesium intake with episodic ataxia type 1?
Not directly. So not directly. Of course, magnesium, but this is I don't talk as an expert, but clearly magnesium can be helpful to lower the activity of calcium channels specifically, so they can be helpful to reduce this hyperexcitability. So I would imagine that if this was the question, I would imagine that magnesium might be helpful.
Well,
Supplement with magnesium, but I don't talk as an expert in this case.
Yeah, so something to verify with a healthcare professional, but thank you for sharing that insight.
Yes.
And then we
Definitely.
have another attendee who's asking, how can electromyography help us or give us details about episodic ataxia?
Well, essentially, this twitching is quite typical of episodic ataxia type 1, but sometimes the twitching is so strong that you can see it. Some other times you cannot see it, but if you use electrical techniques, you can essentially detect it. And this is useful because if this is present, then we can say, well, most likely there is episodic ataxia type 1. Like if instead, if we see nystagmus, we can say, well, we have episodic ataxia type 2.
That's very helpful to know. And then we are at the top of the hour. So thank you so much, Dr. Cingolani, for a fantastic talk. That was jam-packed full of information. So thank you for walking us through it.
Thank you for inviting me. It was a pleasure.
And then for everyone who's attending today, I hope you found something useful from this session. You learned a little bit more about episodic ataxia type 1 and 2. So wherever you are in the world, good morning, good afternoon, good evening. Hope you have a wonderful rest of your day. Take care, folks.